The discovery and the commercialization of these drugs is unlike any other part of the pharmaceutical industry.
These are for treatments that there are no other therapies, and without them, many of these patients would die.
Can it be done? Can it be done safely? Can it be done effectively?
There's typically a trade-off between scale and risk. In this particular environment, the risk element must take precedence.
Understanding what the true critical quality attributes of these products are is still early on. Hard to develop a standard if you don't actually know what you're looking for.
The global pharmaceutical industry is constantly innovating, looking forward to discover the next class of therapies to treat and cure patients around the world. Cell and gene therapies, which fall into the broader category of drugs known as Advanced Therapy Medicinal Products, or ATMPs, have emerged as an incredibly promising class of treatments that have shown the ability to treat and even cure many individuals with diseases for which there was little to no hope. Cell and gene therapies work by substituting the missing protein or cells causing the disease's symptoms. Cell therapy treats diseases by restoring or altering certain sets of cells, or by using cells to carry a therapy through the body. Gene therapy operates differently by replacing, inactivating, or introducing genes into cells, either inside the body or outside of the body. Some therapies are considered both cell and gene therapies. These therapies work by modifying genes in specific types of cells and inserting them into the body. Current market forecasts predict the cell and gene therapy market to grow by almost $10 billion by 2026, a compound annual growth rate of more than 20%. American Pharmaceutical Review and Pharmaceutical Outsourcing magazine spoke with industry experts to learn how cell and gene therapies are changing the healthcare landscape and what needs to be done to bring these advanced therapies to patients. As with anything new in the industry, there is a learning curve, and acceptance of new technologies takes time. Currently, there is a lot of interest in these therapies, but also some hesitation as the industry considers the costs, technology, and regulatory challenges of these therapies.
If you only go back, say, 15 years, cell and gene therapy was still in the space of academic research. It was promising, but there were no actual products. And it was only really when the first CAR T therapies were manufactured in the very early days of the cell and gene therapy industry, so going back, say, 10 years, people thought, we're actually making a breakthrough here. And 10 years on, we now begin to see patients, those early patients, who are now still cancer-free, and by clinical definition, that actually constitutes a cure. So we're actually beginning to talk about curative therapies for cancer, which we would never dreamed of just 10 years ago.
I liken, where we are right now in cell and gene therapies, where we were 40 years ago when we were talking about antibody therapeutics. We're just scratching the surface of what we can do with them, how to manufacture them cost effectively. And then once that happens, many, many more treatments, I think, are going to be amenable to using these novel technologies to treat them in the future.
Cell and gene therapy is very new, it's very exciting, but I think that it is being widely embraced, and you can see that across a couple of different reasons. One, you can see the number of companies getting involved, and two, you can see the number of new products that have started filing INDs and things with the FDA. So you can see a lot of growth in this industry. It is still very new, but there's a lot of growth and a lot of exciting things going on in cell and gene therapy.
There are the cell-based gene therapies that are being used to treat people with certain types of blood cancers. And then there are gene therapies that are addressing disorders that were previously unaddressable by providing therapies, for instance, for children with rare disorders. And they're increasingly finding their way into the treatment of adults with various hematologic cancers.
The emergence of cell and gene therapy, no doubt, has taken the market by storm. Since the commercialization of the first chimeric antigen receptor T-cell therapy in 2017, there has been not a trade show or conference where cell and gene therapy didn't headline. The emergence of these new modalities has created a lot of new challenges for the market and created a lot of new challenges for the pharmaceutical landscape. Where once large pharmaceutical companies predominated the market in various modalities and therapeutics, we see the emergence of small biopharmaceutical companies leveraging very niche experience to solve unique problems.
There is a significant adoption in the space to the tune of over $22 billion in direct investment in the cell and gene space last year alone, and $19 billion the year prior. Within cell and gene therapy, the vast majority of companies are extremely well funded, have very deep pipelines and extensive relationships from a partnership basis with large pharmas and biotechs. And so the adoption is a pivotal component of almost every company.
This industry is an industry of attrition, where you look at a disease state, you test that disease state, you see if the proof of concept is there. And if it is, then of course you move forward, which that's what the sponsors desire to do, is to move that therapy forward into whether it's gene therapy or whether it's cell therapy, and to move from the research phase into the clinic phase as quickly as they can and as efficiently as they can.
Cell and gene therapies require a completely different model for development and manufacturing compared to more traditional large and small molecule products. Many of these critical industry issues are still being perfected and streamlined as the industry is realizing the potential of these products to treat patients who might have been considered untreatable.
So if you look at a typical process that's been commercial for 10, 20 years, I mean, a lot of the different failure modes. And you've also been able to use the tenure of the people that are already there to train the new people that are coming in, to build those libraries, and make sure that you have the appropriate baseline knowledge to be successful in those situations. When you get into clinical products, and especially clinical products in the cell and gene therapy world, we may have a process that is completely off the beaten path of where we normally walk to. So we're having to learn and train at the same time.
There are a variety of things for cell-based gene therapies for the manufacture of chimeric antigen receptor T cells that involve quite a complex chain of events. So when autologous T cells are made that are manipulated genetically, they actually have to be taken from the patient who is ultimately to receive them. They're harvested by something called apheresis. That's one piece of it. Then they're sent to a manufacturing site for cell-based gene therapies. Then there's a selection process, so you have just the cells you want to expand. You then expand those cells, you put a gene of interest into them. Most places are using a lentiviral vector, although other approaches are also being used. After that portion is done, they have to be expanded, so you have a larger number of cells. They're then formulated. There's some type of quality assurance testing that's done as they're formulated, and then they're given back to the patient ultimately. That's a lot of different steps, and each of those manufacturing steps can have variables.
The starting material is inherently variable. People are different, the genetic makeup are different between peoples. Now when you're starting with a variable starting material, it's very, very difficult to make a standard finished product, especially when it has to go through various rounds of manufacturing and the supply chain and the administration, which are all points of variation. So that's one of the biggest challenges, is how the industry tries to create a standard product.
With the introduction of gene therapy, we have now to include areas such as virology and viral clearance or viral safety, which historically have been secluded, and therefore were not run in conjunction with any of the other release panel. And that in essence forced us to increase the skill set as well as the expertise needed for running one single release panel on one single product. In a similar manner, the instrumentation requested to perform the full release panel on gene therapy, for example, have demanded us to really look into instruments that typically belong to the protein characterization area. And moving those instruments from the research and development to the GMP with all the constraints of data integrity posed difficulties. The typical skill set to run this instrument but also interpret data coming out of them is not one that is typically found in QC groups. For some cell therapy products, release testing has to happen in a matter of hours, This is radically different from what we had to do to release protein. The other issue that is very, very typical and specific to gene therapy and cell therapy is the scarcity of the material. We never have enough material to do what we want to do at any given point in time. So we had to actually take different courses of action and also rethink entirely the experimental design of our validation. Then the last main issue is the few number of lots that are going to ever be manufactured throughout the entire product lifecycle. For the innovator, that is very difficult to actually just seclude, generate, or even retain one drug substance or drug product lot to create a reference standard for that product, which trickles down to the analytical group as having difficulties to rethink the assay control strategy or the system suitability strategy. The team at Eurofins came together to find a creative and agile way to provide still the same level of GMP compliance, the same expertise, but in a completely different environment, an environment that was much more suited for the very specific demands of those products.
So gene therapy is a gene replacement technology. Either the gene is missing or it's broken. Then of course there are editing technologies where maybe you just go in and fix one base. But researchers may take years, decades to discover the key. Once the key is discovered, then organizations like Catalent and others can then manufacture the therapy. Some of the other challenges are cell culture. So of course we're making the vector of interest, but you can inadvertently grow adventitious agents, things that may actually harm the patient. So there are enormous kinds of efforts, for example, single-use technology, to ensure that there's not cross-contamination from one production to another, or of a culture with an agent that may come in environmentally.
At the end of the day, you want a safe, efficient gene editing step, but also one where the cells are viable in the end, and that is scalable enough to manufacture this in a batch or campaign format. So we use a proprietary electroporation device that we have fine-tuned exactly for our gene editing step in order not only to reach exquisite levels of gene editing efficiency, lack of off-target effect, but also excellent viabilities for cells in the downstream.
The difference between cell and gene therapies and other therapies is we have someone waiting on that patient, a particular individual. So it's not like I can make a wide variety of pills or have a stockage of medicine and have that waiting for when a customer or a patient needs it. We need to have the ability to react quickly to new patients and be able to treat them because these are life-threatening diseases.
It's speed to clinic. A lot of the indicators and a lot of the projects have to be delivered in a fresh state. There's very little time between the time that it is put in its final container enclosure to then move it to the clinic so that it can be administered. A lot of times you're not even given the time to complete all the testing, and sometimes it's even agreed to as moving it at risk. However, that doesn't put the patient in jeopardy. What has happened before that time is that a lot of in-process testing, a lot of indicators, and a lot of analytics is done before the final C of A to grade or judge or metric how well is the process going, moving from, say, the beginning of the process to the end of the process to the final formulation of the vaccine before it moves to the clinic.
Because these drugs are so unusual, they're actually manipulating the patient's own immune system, or maybe the genetic makeup in the case of gene therapies. Nobody really understands what the long-term effects are, and that's why the regulators like the FDA and the EMA are now stating these patients have to be followed up. In the case of gene therapies, up to 15 years post-treatment. The problem is, how do you follow up patients after 15 years? People will move, they'll get married, they'll change their names. If they recover, they might not want to go back to hospital for follow-up visits. So that's another challenging aspect of how we follow up these patients. And those are just some of the challenges. So it gives an indication of some of the issues that the industry is facing at the moment.
Cell and gene therapies require very specific technologies in order to develop, manufacture, and ship these products to patients. In many cases, time is of the essence, as these treatments are sometimes the last hope for patients with life-threatening diseases.
Just as the contract development and manufacturing space has increased to meet the needs of the cell and gene therapy industry, so have we at Eurofins. We've expanded our contract testing labs. We've built hundreds of thousands of new square feet of laboratory to support this industry. We've created duplicate and replicate efforts in different parts of the world, building test capabilities in Europe just as we have in North America, building redundancy in North America. Our goal is to move just as nimbly and quickly as the industry.
We've considered things like pass-throughs and individual BSCs for each of the tests that we're performing. In addition, as we were looking at our space, we really considered how will samples move through our space and how will people move through our space. All of these things are helping us to deliver high quality testing results in an efficient way.
Development and manufacturing can be made simple if you have single-use, mass-produced, readily available consumables. It becomes a little bit more complex when you add in elements such as viral vectors, plasmids, small target population cells, and costly reagents that come in quantitatively small sizes. When that happens and those elements are essential to a novel therapeutic, you get a drive up in cost. Additionally, it's more supported for your process to be more closed and not have so much potential to have human error. So you have automation technology that comes into play in order to streamline the process and to ensure good production. And then with those automated technologies comes programs and protocols that come with the additional fees such as software and licensing. So cost, the availability of very expensive, very rare essentials to a process, all of those things are variables that can cause it to be a little bit more difficult.
There's not one magic piece of technology that's going to streamline and enable the process. But at Cryoport, we are looking at whether it's the logistics, using shipping devices and technology that is innovative and helps protect the product, or whether it's our bioservices capabilities, enabling technology for the GMP-controlled storage of material, packaging and labeling technology at cryogenic temperatures, or the management of clinical trials throughout the world.
The scalability aspect and automation-related considerations for scale in the manufacturing environment, in the processing environment, in the collection environment, even in the transportation environment, all must be considered and must be considered without compromising the integrity of the product or introducing risk into the system. Obviously, there's typically a trade-off between scale and risk. In this particular environment, the risk element must take precedence and be addressed in conjunction with the scalability of the different processes that are associated with the manufacture and distribution of these products.
Some different ways that we can look at scalability is what we're doing here at Lonza with the Cocoon platform, that we can have trees of cocoons and multiple processes going in a closed system at the same time, which allows us to treat and produce products for more patients simultaneously. Additionally, there's more and more development work going from autologous cell therapy towards allogeneic therapies, which will allow us to have more patients treated for one process run.
So the beauty of allogeneic CAR-T products is they can be manufactured at scale, and a scale much larger than autologous product, and stored ahead of time. Our products are cryogenically stored, and we have run stability studies showing that they can be stable for periods of years. There is no constraint on shipment and timing because everything can be organized in a batch fashion and sent to clinical centers before a patient is even identified for treatment. So we believe that allogeneic is going to solve a number of the shipment issues and turnaround times that have been seen with autologous product, and that they can replicate what has been seen in other drug products such as antibodies or small molecules.
Partnering with your vendor is critical. You need to work through the engineering aspects to ensure that you can be as closed as possible. You'll hear a term in the industry of being 100% functionally closed versus being 100% closed. Here at Medica, we're moving to 100% close because we think that it's critical. It also de-risks our process. It increases the potential for safety of the product itself. It also ensures that what we deliver at the end is safe, sterile, and effective.
The development, manufacturing, and shipping of cell and gene therapies requires a very specific set of technologies, expertise, and experience. It is increasingly evident that to successfully provide such an advanced therapy to patients requires a combination of service, technology, and raw material providers working in conjunction with each other.
Increasingly, there are providers who are developing reagents, raw materials that one needs to make these products that are well-defined, that meet the quality standards that one needs for good manufacturing practices. So that's an important piece. And then there are service providers like contract manufacturing organizations that are starting to come along that will be able to help smaller companies or academic investigators make these therapies without having to start from scratch learning the entire process. So it's actually going to be very important for the growth of the industry potentially to see these contract manufacturers come in who can help the investigator who is trying to develop something. They may have the intellectual idea of how to do something, but they may not understand all of the aspects of the complexities of the manufacturing. And having these contract manufacturers may help speed these things along, as opposed to having the academic investigator have to learn by trial and error or relearn what others have learned about the manufacturing process.
There's a lot of different industries that are involved in the manufacturing of these types of products, and no one entity can be the sole provider for all these. So as a result, we have to work in partnership with vendors who supply the key critical raw materials as well as the equipment needed to manufacture it, testing laboratories that have unique proprietary testing methods used to identify and to characterize the molecules. And then there's CDMOs like ourselves who manufacture it. And then you have the clinicians that actually have to implement it, as well as the principal investigators who develop the product to begin with. So it's a lot of different parts that come together in order to make one of these therapies.
The main challenge for CDMOs today has been to meet the timelines and the requirement of these companies, especially in an environment where supply chains have been severely hit, in particular by the COVID-19 crisis. And so this is today extremely difficult, and there's a lot of competitions from drug developers to access these manufacturing slots in order to progress their products fast into the clinic.
With the unique landscape of cell and gene therapy comes a new need to invest in technology that meets the needs of these modalities. Traditional analytical technology is not always the best solution. For instance, traditional sterility does not meet the incredible vein-to-vein turnaround time for autologous cell therapy. The control strategy that meets the needs of these modalities is more important than ever, and it requires unique thinking and innovating in order to satisfy client and healthcare needs. That isn't to say that traditional analytical work isn't here to stay. We talk a lot in our area about design of experiment and quality by design. These ideas are more important than ever. We need to be able to manage the timeline for testing. We need robust methods that can adapt to changing landscapes of materials, where you have inputs from different patient autologous treatments that can vary, matrices that don't always match. Having methods that meet the requirements of ICH, but also meet the requirements of our clients and their patients is critical.
Some of the things that we've been looking at to address this particular aspect is really driving next generation technology to provide more visibility during transportation, to more control over addressing risk while product is in distribution. For example, eliminating the risk of carrier mishandling on a product, providing better data chains that have artificial intelligence to look at risk associated with drug product distribution, looking at automation to address the scalability issue, as well as building out infrastructure. And so there's a lot of investment going into the space that is really focused around manufacturing capacity, significant investment in building out infrastructure related to drug product storage, as well as distribution.
Our raw material providers and our logistics providers are absolutely crucial in this entire process when it comes to CGT. So when we get a leukopakins blood product from a patient, it's incredibly important that we're ready to process right when we receive it. And that means that we have all of the raw materials ready and that we have everything we need ready to go when that happens. And so a lot of times that means a lot of coordination and working with our raw material providers so that if something happens or if we need a rush supply, that they're willing to get that to us as quickly as possible so that we can keep the product going for our patients.
We are investing in a training facility. We are investing in bringing in other platforms to help us do that quicker so that we're not having to build everything ourselves. We're relying on things that other groups have built and we're building off of that. We're also moving to hands-on training a lot more. Getting the opportunity to practice with a piece of equipment in a live setting that's not in a GMP setting so that you can fail, you can learn from that failure and you can move forward and it's not going to ultimately affect the patient.
First and foremost, the CDMO and your vendors need to partner. What we've seen due to what has happened to the global supply chain with COVID is that everyone has run out of raw materials, components. It's so critical for every process that we do to establish those relationships, those partnerships, becoming a key account with the suppliers of those raw materials, and then to project out forecast, to look at ways we can hold those raw materials without them expiring. If you don't have those types of relationships, then you may be dealing with a lead time that causes us as the CDMO not to be able to start on time. Now all parties suffer due to the fact of that long lead time item.
The pharmaceutical industry is perhaps the most regulated global industry. For a product to be released to patients, it has to pass strict regulatory requirements and a final review by healthcare agencies. Frequently, when regulatory and standard-setting agencies and organizations see the benefits to new products, they provide guidance to companies to help them develop cutting-edge pharmaceuticals.
This is new. Everybody's learning together. So there are aspects of the manufacturing of cell and gene therapies that even the FDA don't understand, and they admit they don't understand. However, there are some areas where standards are beginning to emerge where they never existed before. So one example of that is actually the transportation of cellular material for therapeutic purposes. Up until recently, there were no standards controlling this. Everybody just tried to do the best they could. They followed maybe some best practice, but there was huge variation. So now the FDA and USP and the standards coordinating body and industry like Cryoport have been involved in creating new standards that have now been adopted by ISO, so these are now international standards, in the form of a new standard, which is ISO 21973, which starts to address some of the challenges in the lack of standardization in transportation. It gives an indication that standardization is coming into this industry, and that's one thing that will help with the variability of these products.
The FDA and USP and other standard-setting organizations have absolutely provided guidance on supporting this industry. The unique challenge is that guidance is new. Following that guidance and adapting to processes and systems that were developed for a different regulatory landscape makes this very challenging. Eurofins is able to leverage massive experience across the industry within this modality to support quality control testing that will satisfy regulatory scrutiny.
What is the best way to ensure quality control and safety as we're performing testing? We've been fortunate enough to be able to leverage many webinars, seminars, and conferences, in addition to leveraging FDA resources that are available on the topic. We are working closely with our partners through industry, and we are leveraging information that we have learned on past therapies to ensure that we are meeting the demands of cell and gene therapy.
The regulations evolve as the technology evolves. So what we do here at Lonza is we're actually working with the regulators to make sure that we understand what's coming up, what regulations are coming that could affect us, but also that the regulators understand the implications of the guidance that they're providing and also what guidance is needed so that we can make sure that we're working together to have a safe and effective product for the patients.
you have history on your side when you work in some of the other disciplines of pharmaceuticals. I think we're still trying to answer questions, the regulatory bodies are still trying to answer questions, and luckily there's a very distinct and strong collaboration there to get it right, to make sure these products are safe and efficacious, and to make sure that we are putting the right measures and regulations in place to be successful.
Regulatory organizations such as FDA and EMA recognize the growing importance of gene-edited and CAR-T products. This is why they have published critical guidances over the last couple of years. One guidance in 2021 from EMA on gene-edited products, and 2 guidances from FDA on CAR-T and human genome-edited products. These guidances are particularly important for developers such as us because they provide the level of quality required by agencies for future approval of the therapies, as well as the level of flexibility they have in an ever-evolving field.
What I observe and find, the FDA and EMA are really looking to be partners in getting these therapies to market. Of course, safety is paramount and efficacy is critical, but once these things are apparent and demonstrated, the regulators are really extraordinarily strong partners because at the end are patients who, when they receive these treatments, could actually be cured, which is just really amazing. So I think we all understand that, and when we work together, we just get to more patients.
At FDA, we've put forth some general guidance around the development of cell therapies. We anticipate getting out more guidance in the not too distant future, specifically related to the development of chimeric antigen receptor T cells. We think that'll help the industry. There are other organizations like the Standards Coordinating Body. Those organizations are trying to help understand particular aspects of quality control, for instance, or standards that may help manufacturers as they develop these. Part of the issue that's challenging right now is understanding what the true critical quality attributes of these products are is still early on. So as we get more experience, hopefully we'll start to understand what truly are the critical quality attributes that you can develop standards around. Hard to develop a standard if you don't actually know what you're looking for. So hopefully that's going to be something that will become clearer. I know we're working towards that at FDA, and I know others outside of FDA are doing the same.
There is no doubt cell and gene therapies hold incredible promise to help many patients with devastating diseases. Many innovator companies, along with technology and service providers, are pushing the envelope as quickly as possible to bring these treatments to patients in need.
Can it be done? Can it be done safely? Can it be done effectively? If those answers to those questions are yes, then of course the industry embraces that technology. There's many that are out there today. And there's many more in the pipeline that will soon be here that this industry can take full advantage of. At the end of the day, we all need to keep in mind that these new technologies and the research associated with them, getting through your proof of concept, all of that is phenomenal. That's great news. But where we need to also never take our eyes off of is that we work for patients. We work to cure disease states that have not to date had the cure rate that potentially some of these new technologies will bring to the table.
In the future, we'll see cell and gene therapies being used not for the last line of treatment when everything has failed, but we'll see them as first-line therapies. We're even seeing now CAR-T therapies moving from third or fourth line treatments up to second line. And that's an indication of confidence, growing confidence. But it doesn't come without its challenges. So even if you look in the clinical environment, even now with the limited number of cell and gene therapies, hospitals and clinics are complaining they don't have enough trained staff who understand how to administer the drugs, how to monitor patients in the future when these drugs become commonplace, how are hospitals and clinics going to be able to cope with that? So these are some of the unknown challenges of the future. But Cryoport, as well as other technology companies, we are striving to develop the next technology that will help this paradigm shift to cell and gene therapies in the future.
I think these therapies are eventually going to be at the forefront. Right now, CGT is still pretty new. I see a bright future. What we are working on is making the therapies more affordable, more readily available. So going from autologous, which means from patient back to patient, into more of an allo process where it is off the shelf ready and available as a therapy. And so that is the new wave in CGT, is trying to get more allo products, but auto is still very useful, very productive, and doing very well.
I think when it comes to cell and gene therapies, everyone understands the importance of what we're doing here. So it does take a lot of different players, from us as a contract manufacturer working with our customers, them working with the clinics and with the patients, raw material suppliers, from beginning to end. There's a lot of different people that are all involved, making sure that this therapy can come together for the patient. And so it's one of those things that requires a lot of coordination, but when everyone understands the goal in mind and what we're trying to achieve, it's something that we can all come together to do successfully. There's a lot of exciting things going on. There's a lot of growth in this business, and I think the more and more we see products go from the phases of clinical production into commercial production, the more we're going to get involvement in the industry and the better that it's going to be.
I strongly believe there is a bright future ahead for CAR T-cell therapy. The first step was really the proof of concept with the approval of the first autologous therapies, which are now moving up the lines of treatment, in particular in lymphoma and potentially in myeloma, which will allow them to benefit a broader patient population. The second major step is going to be the development of allogeneic CAR T-cell therapy, with the first approval of an allogeneic CAR T product in the coming years, which we hope will come from our portfolio. Finally, the next frontier is going to be for solid tumors, which together represent more than 90% of human cancers, but this will only be possible through more complex gene editing and cellular engineering of the CAR T product. But all in all, I strongly believe that the allogeneic approach has the potential to bring cells to become true industrial pharmaceutical drugs, paving the way for a revolution very close to the one that was seen with antibodies in the 80s and 90s.
If the industry can come together and take their massive knowledge in this space and their aggregate experience, we can do better when it comes to managing these materials. It is not lost on us as a contract testing lab that many of our clients have to duplicate the same efforts. This goes right back to pharmacopoeia. How do we as an industry come together to support test methods that we know to be good and trust, that meet the quality requirements that we have, and lower the overall cost of goods for not just the therapies of today, but the therapies of tomorrow? We know we'll see these materials for a long time, and being able to work and collaborate as an industry is critical for us to be successful.
The fact is that most of these diseases, are affecting children. And when you think about the ability to treat a child, to cure its disease, and we're not talking about palliative care, we're talking about actually curing a disease, making it completely vanish, or in hopefully the optimal scenario, treating before any symptoms ever develop, and thus the patient never ever sees any of the consequences. You're curing not just that child, you're curing a family. That disease, if it were to progress, is going to affect everyone in that family, and to think that with one single dose, we can make it all go away, to me, that's a miracle.